Hello, Grateful, Mr. Stole here from your tutors. So today we are looking into protein synthesis. And with protein synthesis, you must know that this is the manufacturing of proteins, number one. And also you must know that it consists of two stages or two steps, which is transcription, the first step or the first stage, then translation. Okay, so it consists of transcription and translation. So another thing that you need to know is that the location where each of these processes takes place. So transcription takes place in the nucleus, okay? So it takes place in the nucleus, and translation is the one outside here, is taking place what you call on the cytoplasm. So cytoplasm, ribosomes. Okay, so it's actually taking place on the ribosomes, what called inside the cytoplasm. Now, then another thing also, you must be able to know that when we're talking about transcription, we are saying messenger RNA is copying the code. It's copying the code, what call this one, obtain the code for synthesis of specific protein. So where is the code coming from? The code is coming from the DNA. So you can see that our DNA is double helix, it's coiled, and what needs to happen there, first thing is that it has to unwind, and when it unwinds, a weak hydrogen bond breaks, but only a portion of it, as you can see here, only a portion of it, only a portion here. Let me just use another color here. Only a portion of it is actually what we call being copied here. And what do we call this section here? We call this a gene. Okay, this is a gene. So a gene code for a specific protein. So your DNA is big. It cannot leave the nucleus. That's why it has to send the message with the messenger RNA. Now, so you must know the process there, that after the weak hydrogen bond breaks, your DNA unzip, as you can see, then free nucleotides. So there are free nucleotides lying around in the nucleoplasm. Those are the ones that will come and form the complementary messenger RNA. Okay, then it's through complementary base pairing, remember? So it has to be A pairs with U. Why are we having U now? Because now we are talking of RNA now. Because we are on RNA, we no longer have thymine. Then C, what called pairs with G, then that's it. So you must know the process, you must know these steps, and you must not forget that only one strand is used as a template. So here, after it have got what called it opened, only one strand is only used as a template. The other strand is not used as a template. If you check with replication, both strands are acting as templates. Okay, now the messenger RNA is formed. What does it do? It leaves via what we call the nuclear pore. So this is the nuclear pore. It via the nuclear pore. Where does it go? It goes to the ribosomes. What is the ribosome? This is the site for protein synthesis. Site for protein. Okay, now, so once it reaches the ribosome, it lies, what call it, aligns itself with the ribosome. Then what we have in the cytoplasm here, we have what called transfer RNA. Transfer RNA, which is carrying amino acid. It's carrying amino acid, and when it's carrying amino acid, on other hands, it have got triplet bases, and these triplet bases, we call them anticodons. So for example, here we can say A, U, C. So in this case here, this is the what? This is the anticodon. So anticodon is triplet. We are referring to triplet transfer RNA basis. Then here, what we have, the complementary basis for anticodon is the what called the codon. So here we can say, let's say we have A, U, C. Then here we have what called U, A, G. Okay, now this UAG here, we call it the codon basis. We call it the codon, called codon. Now, what is codon? Codon are the triplet messenger RNA bases. So 3 of them, one codon consists of 3 bases. So one codon consists of 3 bases, and one anticodon also consists of 3 bases. That's why we're saying triplet. Okay. Now, what happens during the translation? We are saying messenger RNA leaves the nucleus through the nuclear pore. What does it do? It comes align itself with the ribosomes. Then we will transfer RNA that brings in amino acids to the ribosomes. Once it brings the amino acid to the ribosomes, you can start adjacent transfer amino acids that will form what called a peptide bond. So these are amino acids, and what it form, they form a bond, and the bond, that bond, we call it peptide bond. Peptide bond. And the peptide bond is formed between amino acids. Okay, so you must always remember that it's formed between amino acids. So you can see that now we are forming an amino acid chain, which we call the protein, or we can say it's a polypeptide, forms a polypeptide chain. Polypeptide chain. Okay, so that's what we have. So this is a process. You must always remember the process of translation and transcription. But in most cases, transcription is being asked in your exams. But you must remember also what happens when you're out, when the messenger RNA reaches the ribosomes. Okay, now that long chain of amino acid is our protein, and it performs specific functions. It performs specific functions. Now let's get to something also to recall when you're looking at replication and transcription. Now, with replication, you can see that 2 DNA molecules are formed. 2 DNA molecules are formed. 2 identical DNA molecules are formed. So here, 2 identical DNA molecules. Now, these are formed. What about when it comes to transcription? Transcription, what we see that the product only is the messenger RNA. Okay, messenger RNA, and the, what is it? And here, another thing here, we are saying 2 strands act as templates. So here, 2 strands, so 1 strand and number 2, they act as templates. So 2 strands act as templates. But on the transcription, only 1 strand act as a template. So it's something that you must indicate that in your explanation, especially when you are explaining the process of transcription, you must indicate that 1 strand act as a template. Okay, but the rest, you can see that is the same DNA. It has to unwind DNA, it has to unzip, and the free nucleotides are always being used to build the complementary strand. Okay, now let's look into questions that comes pertaining protein synthesis as well. So this case now, we're talking of gene mutation. Now, when we talk of gene mutation, these are changes in the DNA sequence, changes that changes in the DNA sequence. So here the definition is there. Sorry about that. Changes in the DNA sequence. So what we are saying here that, let's say for example here, a section of messenger RNA has the following sequence, base sequence, and is read from left to right. So GAU, it goes on like that. And what do you call this? These are they are codon. So these are the codon. Okay. Now, a mutation occurred which resulted in the following base sequence on the messenger RNA molecule. And what we see, you can see that if you start from the first one here, GAU, this GAU, CUC, GAC, ACG, AGC, AGU. So you can now see that the mutation is here. There are changes here. And what are the changes? C, which is cytosine, is replaced with with U, which is uracil. So the mutation here, C, was replaced with U on the 4th codon ACG. Now, what effect does it have? What does this mutation have on the resulting protein? This is the common question that comes asking you, what effect does it have on the resulting protein? Now, what you can see is that we have got AGC, then we have got AGU. you go to your table that you are given. We look for AGC. AGC is here. And what does it code for? It code for serine. Then AGU is here. What does it code for? It code for for serine. Now, in this case here, we can now see that there is no change in the amino acid that is going to be formed. Okay, so now what you do now, you're going to say, okay, both, so you can see that it codes for the same amino acid. So we're saying both A, GC and AGU code for same amino acid. Now, when it codes for the same amino acid, it means that there is no change. The amino acid sequence will not change. That's the good thing. So you must mention that amino acid sequence will not change. Therefore, there will be no effect on the protein formed. So we are saying that it will form same protein. So here, same protein is being formed. Okay, now, so now we can move on to the next one. Now here we are also being asked here the best triplet, the DNA base triplet 1, 2, 3 below is read from left to right. So we've got GTC, AAG, and CCT. A mutation occurred which resulted in the following base sequence on the DNA molecule, which is GTC, TAG. Now you can see that the TA was replaced with the T. So A was replaced with T and CCT remains the same. Describe the mutation that takes place. So in the DNA triplet, triplet 2, okay, the first adenine was replaced with T, which is thymine, okay? Now, what effect does it have on the protein that is formed? Now, as you can see here, we are saying your AAG, the codon. So the codon will change. So the codon now is now here the codon here will be, because this is the DNA, so the codon there will be AUC. The codon now is AUC instead of, so instead of what? Instead of UUC, instead of UUC. Now, when we go to the table now, we now look for the codons, because this is showing the codon. So as you can see here, the codon AUC code for isoleucine. Then the UUC code for phenylalanine. Now, can you see that? So these two are different. So now a different amino acid, isoleucine, will be coded for instead of phenylalanine. So in this case now, we are saying a new amino acid is actually introduced in the sequence. Now, when we have that, it means that amino acid sequence will change. So amino acid sequence will change, and when it changes, a different protein will form. A different protein will form. Once we form a different protein, it means that the protein will perform different functions. Different functions. Okay, so this is what you need to state. So you must always know that when you ask such questions, you must mention, you must look for the amino acid that is being formed. Is it a different amino acid? Is it the same amino acid? If it's the same amino acid, there won't be different, there won't be changes on the protein formed. But if there is there is a change in the amino acid, then the protein that will be formed will be different, and it will be performing different functions. So we call this harmful mutation. Harmful mutation. And the previous one here is a harmless mutation. This is a harmless mutation. Okay, so another thing that I just need to add here, another thing that you need to add here is that the mutation that types of mutation that we have what mutation that are point mutation. So point mutation is just affecting a triplet base, a triplet, what called one codon. Okay, it's just apply one triplet base. So point mutation, point mutation. So in this case, the a good example here is where we have got, like, for example, here, if we replace, so if we replace a C, we can that replace C was replaced with U. Okay, so here we call that substitution. So substitution. Then another thing that can also happen, we can have a swap to what is it, two bases, let's say from A G C to C G A. So in that case, there we are saying it's an inversion. Okay, so substitution and inversion cause a point mutation because just affect one triplet base. What about frame shift mutation? Frame shift, the one that cause a frame shift. A frame shift now, it means that if one is deleted, so let's say the good example here is deletion and addition, insertion. So if you insert, if you delete, let's say you delete, if you delete any amino, any base there, let's say we delete a G. If you delete G, it means that we are going to have, in this case here, we are going to have A, U, A, because the A will go there. Then the last one will be what we have C and C. That now is causing a frame shift because it needs to fill up the gap that was that was created there. Because once we remove G, it means that we're left with a gap. So A has to move and replace that gap. So that's what we call the frame shift, that is being caused here. So it's a deletion. So if one base is deleted, it means that it's going to cause a frame shift in the sequence of the in the base sequence. What about insertion? So if we are to come here and insert, let's say here you insert maybe let's say you insert A. So it means that you're going to have A, U, A. Then we are going to have G, A, C. Then the last one will be C. And the C does not code for any amino acid. And also these two C's cannot code for any amino acid. So you can see now how the mutation will affect your protein that's going to be formed. But this now these ones here will cause a harmful mutation because now you're going to have a frame shift. You're going to have maybe a less number of amino acid that was supposed to be there if there is a deletion. And if there's an insertion, you're going to have another, depending on how many amino acid how many bases were inserted, but you can now end up having extra amino acid that were not in the sequence, but resulting again in a different protein, which is part of the harmful mutation that we mentioned. Okay, now I think you guys, you are sorted now. Thank you so much. Until next time.